This is our third recording for Chapter 6, and I hopefully I can get done with it in this recording. But anyway, we are now discussing the cytoskeleton, which consists of 3 different types of molecules. They're really, they're types of protein fibers. So we have the thickest protein fibers or filaments are called, the thick filaments are microtubules. The thin filaments are microfilaments, and the ones that are in the middle thickness are intermediate filaments. All right, there's an animation on slide 68, and this slide just kind of shows you some functions of the cytoskeleton. One of the functions is it makes these tracks, like for the vesicles and other organelles to travel. It's like train tracks kind of, but it produces these tracks inside the cell that kind of guide vesicles and other organelles to their destination. And the way they travel is really kind of cool. All right, so like this is a vesicle that's having to move through the cell, right? Okay, so an ATP has to bind in order for this process to work. But basically, you have this microtubule or thick filament in the microtubules are thick filaments, okay? So you got a microtubule of the cytoskeleton, and then your vesicle attaches to a motor protein because it's actually powered by ATP. And the motor protein like changes position of its little feet structures. You see, it just kind of walks along these tracks until the vesicle reaches its destination. So that's one of the functions of microtubules. And this page just tells you microtubules are the thick filaments. Microfilaments, I do want you to know this, microfilaments are also called actin. And that's going to be important if you ever take anatomy and physiology. Actin is one of the filaments in a muscle fiber that cause muscle contraction. Basically, you have a thick filament called myosin that attaches to actin and pulls on it, and that's what causes a muscle contraction. But in the cell, actin filaments, that's just the name for the microfilaments. They're called actin. Those have, play different roles, and they can play a role in movement. And then you have the intermediate filaments, and they're going to be the middle thickness. You have some functions down here. You can see that all of the protein fibers, all of the members of the cytoskeleton help to maintain the cell shape. So you have to look below that to see what they do that's different from one another. The microfilaments or actin, as well as the thick filaments or microtubules, function in cell motility, in cell movement. So that can be cell internal movement, or it can be movement of the cell, like we saw the paramecium swimming using cilia on the outside. Those cilia are composed of microtubules. Now, microtubules are very important when we get to mitosis. They're going to attach to the chromosomes, and they're going to line them up in the middle, and then they're going to pull them apart during cell division or mitosis. So microtubules, we're going to call them spindle fibers when we get to that section. But microtubules are very important in chromosome movements during cell division, and microtubules also form those tracks to help organelles move, throughout the cell. The actin microfilaments function in changes in the cell shape. They function in muscle contraction. They function in a phenomenon called cytoplasmic streaming that you see in plant cells. I think there's a video in this, there is, in this slideshow that shows you the cytoplasm just flowing or streaming along inside the plant cells. And actin filaments also play a role in cell division in animal cells, and I don't really know I know what they do. Not in the movement of the chromosomes, but in the splitting of the cells. They form this structure that basically cleaves the cell. I don't remember what it's called, but anyway, they help divide the cell in half. That's actin microfilaments. But that even though they function in cell division, make sure you know the difference. Actin microfilaments, the thin filaments, only function in actually splitting the cell in half during cell division. But the microtubules function in the movement of the chromosomes in cell division. All right, then we have the intermediate filaments, and they basically anchor the nucleus and other organelles. They are more of a permanent, they're permanent fixtures in the cell, whereas actin and microtubules can, they can be produced, and then they can be digested, and, they play a role in the cell, but it's not like a permanent role where they're just stuck in the same place all the time, So let's see. I don't think there's anything more here that you need to know that we didn't say already. Microtubules in animal cells, they grow out from the centrosome. A centrosome is like a complex contains a microtubule organizing center in animal cells. And in animal cells, there's also a pair of centrioles in the centrosome, and they are composed of microtubules, 9 triplets of microtubules arranged in a ring. And so plant cells can organize their microtubules without centrosomes and centrioles. So plant cells don't have centrosomes or centrioles. I thought they did have a centrosome. I'm sorry, I said that incorrectly before in the earlier recording. But plant cells don't have a centrosome or centrioles, but they're still able to organize their microtubules. Somehow they do it in a different way. But this is what the centrioles look like. They're composed of, it's like 3 microtubules form a unit, and there's 9 units of 3 microtubules, and they just are organized like this, 2 barrel-shaped yellow structures, and they're kind of perpendicular to one another, and that's what they look like, and they play a role in mitosis. Microtubules they also compose flagella and cilia. So make sure you know that the fibers that form flagella, which is a long whip-like tail, the only place we see that in human cells is sperm. And then cilia, which are little hairs that will be located on the surface of a cell. They can either be used in movement, like they were with that paramecium that we saw that was covered in cilia. Or we have certain cells, like the cells that line our respiratory tract, our trachea, and our large bronchi, are lined with cilia. And what the cilia does is it sweeps, all those little hairs together will sweep in the same direction at the same time. And they'll sweep mucus and trapped particles, substances. They'll sweep all that mucus and trapped particles out of your air, away from your lungs, so your lungs don't get infected. And this is Chlamydomonas, which I can't even tell by looking at this if they have cilia or flagella or what. So I'm telling you what, I'm going to play this if I can. What if you don't want a highlighter? There you go. Okay, so I'm going to play this just so I can see for myself.
Chlamydomonas is a unicellular green algae with 2 anterior flagella and a single cup-shaped chloroplast.
Okay, so you can see the flagella once you start it moving. So that's pretty cool. So it's an algae, but it's a unicellular green algae that can swim. And that was slide number 78. And slide 79 is shows you flagella. And then here's our paramecium. So paramecium that we saw the very first slide in chapter 6. That's slide 80. You can watch it swimming with cilia. Okay, I'm going to move. There's another animation on cilia and flagella, slide 83. All right, microfilaments. we said, are also called actin. They are thin filaments. Microfilaments that function in cellular motility or cellular movement contain myosin as well as actin, and myosin is a microtubule or thick filament. And I explained this to you, that the myosin will attach to the actin, and then it will pull on the actin, and it will cause contractions. So microfilaments, actin filaments can function in cellular motility, contraction, cytoplasmic streaming, which is when you can like watch the cytoplasm streaming or moving along inside of a plant cell. There's a video in a minute. This is a picture of the actin and myosin filaments that are involved in muscle contractions. The actin is red and the myosin is purple. So these little structures on the end of the myosin filaments, they reach up and they attach to the actin. I'm trying to blow this up. It's not letting me for some reason. It's blowing up my bar down here, but good God. Okay, so we have actin, or actin is the microfilaments or thin filaments, and they're red. And then the myosin is the thick filaments or microtubules, and they attach to the actin and pull on it. And then when they pull on it, it moves inward. It moves in the direction you see with the arrows. And that causes a muscle contraction, and it leads to movement. Then we have this little protozoan here. It's not a paramecium, but it's a different protozoan called an amoeba. And the amoeba moves by forming these extensions called pseudopods or false feet. And it is the actin, the microfilaments that we call actin, that help in that formation of pseudopods in cells that move that way. We also have some white blood cells that move that way of our immune system called macrophages, and they phagocytize bacteria. Cytoplasmic streaming you can watch in this video, which was slide number 89. And then slide 90, we're just a little bit more on the intermediate filaments. Intermediate filaments are more permanent cytoskeleton fixtures than the other two classes of, than the microfilaments or the microtubules. So they tend to be permanent and kind of stuck in place rather than, temporary, only produced so they can play a certain role and then they're broken down. Intermediate filaments are more permanent, and they help shape the cell and fix the organelles in place. Plants have a cell wall. Fungi have cell walls. Fungal cells have cell walls. And so plant cells and fungi and even prokaryotic cells have a cell wall. The cell wall of a plant cell is composed of a polysaccharide called cellulose. But it's other chemicals that make the cell wall of fungi and prokaryotes. But anyway, the cell wall of plants is composed of cellulose. Some plants have, like the harder tissue of plants that's tougher, like wood, has secondary cell walls. But anyway, cell walls are just extra protection. And then organisms that are multicellular that have tissues, sometimes their cells are surrounded by tissue called extracellular matrix. An extracellular matrix is made up of glycoproteins, such as collagen, proteoglycans, and fibronectin. So the extracellular matrix is made up of glycoproteins and sometimes other substances that surround those glycoproteins. And the extracellular matrix plays different roles, but here you can see the extracellular matrix has all of these thick filaments. Here are collagen. The little thin green ones are proteoglycans. Fibronectin is attached to some of your, sometimes attached to your plasma membrane. And then you can see microfilaments or actin filaments on the inside of the plasma membrane here. So extracellular matrix is basically the tissue outside of the cells. Plasmodesmata is a special channel that runs through plant cells so that materials can move from one cell to the other through the cell wall. So plasmodesmata are only in plant cells. Their connections, they connect the cells, but they also allow water and small solutes to pass from one cell to the next. Then we have junctions that connect cells. Some are called tight junctions. Tight junctions connect cells to one another and prevent leakage. So we have the cells that line our urinary bladder have tight junctions to prevent urine from leaking out into our tissue fluid. Some cells have desmosomes, which anchor them to other cells. Desmosomes are not like tight junctions because they don't form a tight seal, but they do form a very strong attachment. It's a very, very strong attachment between the cells so that they're not going to pull apart if they undergo some kind of stress or stretching or something like that. Gap junctions are kind of like plasmodesmata, but we find these in animal cells and animal tissues. Gap junctions connect the cells, but they have channels that allow ions to pass from cell to cell. So there's an animation on cell junctions, and that's slide 103. Let's see if we can finish this up. Yeah, I'm pretty sure that this is just extra stuff that you can look at, extra pictures that you can look at, but I think we're done with the primary slides and notes that you need. I don't know why, but this isn't moving forward. But I'm going to stop the recording here because I know the rest of these slides are like just for extra information and extra images.